Hello again. So the second part of photosynthesis is all about the starch test and photosynthesis experiments. Buongiorno. Now, before we look at the starch tests, it's important to remember what photosynthesis is. So photosynthesis is when plants take light energy and that they combine carbon dioxide and water to produce glucose and oxygen. So if we sum that in a little equation, so we said Connor doesn't want granny's organs. So this is what we had. Carbon dioxide plus water produces glucose plus oxygen. Now, the reason we're starting off with this is because if we want to test a plant to see if photosynthesis is taking place, the key to this is the glucose. If it photosynthesizes, it will convert this glucose into starch. And the thing that we know about starch is we can use iodine to test for it. Now you'll remember that iodine goes from yellow-brown to a nice shade of blue-black if photosynthesis is taking place. So this little video is all based around this test for starch. If photosynthesis takes place in a plant, it will produce glucose. The plant will then convert the glucose into starch. We can test for starch using iodine, and if iodine is present, it will go from yellow-brown to blue-black. So when we're doing the starch test, there's four main steps, and they are boil, dip, and test. And you'll see why in a second. So the starch test then is this. Firstly, we boil the leaf in water for 30 seconds. It kills the leaf, stops any reactions, and removes the waxy cuticle. The next thing we do is turn off the Bunsen burner, and that's because ethanol is flammable. And you boil the leaf in ethanol. This removes the chlorophyll. So what you should see is the leaf should go white and the ethanol should go green because all the chlorophyll leaves the leaf and goes into the ethanol. Then we dip the leaf in the warm water to soften it because boiling in the ethanol will get it all crispy. Then we spread the leaf out on a white tile and test for starch using iodine and note any colour change. When you test the leaf for iodine, it's either going to go blue-black, which shows you that starch is present, or it will stay yellow-brown, which shows you that starch is absent. So let's summarise the main steps. Step 1, boil in water. That kills the leaf, stops reactions, and removes the waxy cuticle. Step 2, boil in ethanol, and that removes the chlorophyll. Now, you do that so that you can see the colour change easily later on when you use the iodine. Step 3, dip in water. And that's because boiling in ethanol will make the leaf crispy. And then step 4, test for starch using iodine. There's going to be two results. Starch is present. It will go blue-black. If starch is absent, the iodine will stay yellow-brown. Now, we should say as well here, an important safety feature is turn off the Bunsen And that is because ethanol is flammable. And that's the starch test. Now, it's important to say here that before we actually ever test the leaf or the plant, what we need to do is de-starch the plant. Now, the point of this is that when a plant photosynthesizes, it makes glucose, and from the glucose, it makes starch. But we want to prove that in our experiments, the thing that we are testing is needed. So it's important that we remove all the starch at the start. So to de-starch a plant, you leave the plant in the dark for 48 hours. So it uses up its stored starch. This means that any starch present at the end must have been made in our experiment. So to de-starch a plant, you leave it in the dark for 48 hours, it will use up its stored starch, and this shows that any starch present at the end has been made in our experiment. Now, why would you ever need to use a starch test? Well, you can basically see if things like carbon dioxide, light, and chlorophyll are necessary for photosynthesis, and that's what we're about to look at. So the first experiment is chlorophyll needed for photosynthesis. Now, this seems obvious. Because chlorophyll is needed to trap the light, we all know that yes, chlorophyll is going to be needed. But how can we prove that it's needed? Well, one way that we can do that is by using a leaf like this, where we have a big green bit on the inside and a white bit on the outside. And this is known as a variegated leaf. It's called that because there are white bits and green bits. Now the white bits contain no chlorophyll, and the green bits contain chlorophyll. Did I say that right? So what I was supposed to have said was the white bits contain no chlorophyll and the green bits do contain chlorophyll. So here's what we would need to do to test this leaf. So step one would be to de-starch the leaf. Number two, you take it and you do the starch test. Now, that means that we do all the 4 steps. You boil, boil, dip, and then test. And the results should be the white bits. They've got no chlorophyll, so therefore there should be no photosynthesis taking place there. So there should be no starch. So therefore the iodine stays yellow, brown. And then the green bits, they have chlorophyll. So that part can photosynthesize, so there should be starch present. So the iodine should go blue, black. And that's that test. And this part just shows a diagram of what that would look like. So before testing, we have the green bits which have chlorophyll, the white bits which have no chlorophyll. After testing, the bits that were green go blue-black, and the bits that were white go yellow-brown. Experiment 2 then is light needed for photosynthesis. Now, of course, we know it is. So this time, what you would do is take a leaf, and this time it's all going to be green, so there's no white bits on it. But what you do this time is you take a piece of card or a piece of foil and cover it. So the part here can get light, the part here can get no light, and that's because this is card or foil and it blocks light. So here's what you would do. Step 1, de-starch the leaf or the plant. Step 2, you cover part of the leaf with card or foil. Then you shine a lamp on the plant. And let's say you do that for 24 hours. Remember, you de-starch the plant for 48 hours. Then you test for starch. So you do the 4 steps: boil, boil, dip, test. And number 5 would be the results. So, in fact, let's draw out what it would look like. So if this is our leaf, this part, the bit which got the light, would go blue-black. And this part here would go yellow-brown. So here's why. This part and this part could access light, so therefore photosynthesis could take place. So starch was produced, so the iodine turned blue-black. This part, there was no light, so no photosynthesis could take place, so there's no starch. So the iodine stayed yellow-brown. And that's experiment 2. So this just shows then a leaf that has been left in the white for 24 hours, so it's gone blue-black, which shows us that starch is present. This one has been left in the dark for 24 hours. The iodine's gone yellow-brown, so starch wasn't present. And this one shows a leaf after it's been tested. So this part, which is yellow-brown, had areas which were not exposed to light, so they were covered up. So this would have been covered with a bit of foil, that shape. And this part could get the light, whereas this part would be in the dark. And then this bit shows a cute little leaf where would have been covered with foil in this area. So this area was in the dark, which is why the iodine's yellow-brown, and the rest of it goes blue-black because it was in the light. Now the third experiment, so experiment number 3, is carbon dioxide needed for photosynthesis. And again, yes, because it's part of the equation. So this time what you can do is you get a plant, and oh flip, that's a chunky plant. let's draw a few wee leaves on it. Now, what you could do then is surround it with a plastic bag. What you also put in there is a little solution of sodium hydroxide, or sometimes it's called soda lime. Now this absorbs CO2. So this bag contains no carbon dioxide. You then set up a second plant, and in this plant you have a bag, but this time the solution is sodium hydrogen carbonate. Now this is different. It releases carbon dioxide. So this bag has got loads of CO2. Now here's how we test these bad boys. So you de-starch both plants. Then you leave both plants in the light for 24 hours in their bags. So just to make it clear here, we've got two separate plants and we're going to set these up at the same time. So you de-starch both plants, then you put one plant in the one bag with sodium hydroxide, then you put the second plant in the other bag with sodium hydrogen carbonate, then you leave them in the light. The next thing, you test one leaf from each plant for starch. So again, you do the boil dip test. Now, here's what we would see. The leaf from plant with sodium hydroxide, the one with no CO2, this is what we would see. There is no photosynthesis taking place. And that is because there is no CO2. So it could not make any starch. So therefore the iodine goes or stays yellow brown. On the other hand, the leaf with sodium hydrogen carbonate, it could photosynthesize because there was CO2 present. So therefore it made starch. So the iodine goes blue, black. So just to tie all of this together, we started off looking at the equation for photosynthesis, which was carbon dioxide plus water produces glucose plus oxygen. The glucose gets stored as starch, so we can test for the starch using iodine. If starch is present, it will go blue-black. If it's not present, it will stay yellow-brown. We can test to see if light is needed, because what we can do is cover part of the leaf. We can test to see if carbon dioxide is needed by using sodium hydroxide to remove the carbon dioxide. And we can also see if chlorophyll is needed by using a variegated leaf. And that's the end of this video. Thank you. Actually, I have to say that was a bit of a lie. There is one last little experiment. So the last little experiment is how we can measure the rate of photosynthesis. Now we can do that because in photosynthesis a gas is given off, and that gas is of course oxygen. So what we can do is use a water weed or a pond weed. The most common one is Elodea. And what we can do is we can shine light on it. Ooh, hello. The light will allow the Elodea to photosynthesize, and it will start to produce magical bubbles. Ooh. Now, what we can do is count those number of bubbles per minute. And that will tell us how fast photosynthesis is taking place at different light intensities. Now in this experiment, there's a few things that we'll think about before we look at the method. So firstly, what are we going to change? So how are we going to measure the rate of photosynthesis? Well, what we change is the light intensity. Now we can do that by moving the lamp closer or further away from the Elodea. We're going to measure the number of bubbles produced per minute. Now that will tell us the rate of photosynthesis. So if there are lots of bubbles, that means that there is a high rate. If there are few bubbles, it means there is a low rate or a slow rate of photosynthesis. And to make sure it's a fair test, we'll use the same Elodea, the same apparatus, and we'll have to control the temperature, which we'll come back and we'll think about later on. So this is our method. We set the apparatus up as shown in the diagram with the lamp 10 centimeters away. That is going to be our high light intensity. And that's because if the lamp is very close to the Elodea, there will be a high light intensity or a lot of light reaching the plant. We then count the number of oxygen bubbles produced per minute. Because we're working out a rate here, we need some measurement of a time. We record the results and then we move the lamp to 20 centimeters away. It's important to give the pond weed time to adjust to the new light intensity, and then we count the number of bubbles per minute again. Now, 20 centimeters away is a lower light intensity. We repeat step 4, moving it to 30, 40 and 50 centimeters. So 10 centimeters was where it was the highest light intensity, and 50 centimeters is the lowest light intensity. Now we would repeat the whole experiment and calculate an average, and that makes our experiment more reliable. So the results that we would predict for this is as the light moves further away from the plant, the light intensity will decrease, so the rate of photosynthesis will decrease and less oxygen bubbles will be released per minute. So a lower light intensity equals slower rate of photosynthesis, so less bubbles per minute. So this would show us that the higher the light intensity, or the closer the lamp is, the more energy there would be available for photosynthesis, and so there would be more oxygen produced. Last we note about this experiment, sometimes if you move the lamp closer, it means the experiment is no longer fair. And the reason for this is because if you move the lamp closer, it heats the water, so photosynthesis would happen faster at higher temperatures. Which is right, because if you think about it, in the summertime, when there's more light and it's warmer, that's when plants will usually go fastest. So what you can do to overcome this in this experiment is use a transparent screen in front of the beaker. That means that the light can get through, but the heat doesn't. And that this time is the end of this video. Goodbye.